Good evening, everyone. I'm Young Lee, the president and CEO of the Damon Runyon Cancer Research Foundation, and I'm absolutely delighted to welcome you here this evening. Thank you so much for joining us. We are all here in this room because we've all been touched by cancer as patients, survivors, and family and friends of loved ones who have been affected by the many forms of this disease. We're brought together by the desire and the need to change the statistics and do better with treatment, diagnosis, and prevention of all cancers. So I want to start by thanking our hosts, David and Sandy Marshall, for their generosity and dedication to our mission. As you may know, David serves on the board of directors for both Damon Runyon and the Fox Chase Cancer Center, and we are deeply grateful for all that you do and are delighted to be here together in this amazing space this evening. Thank you also to the board members of both Damon Runyon and Fox Chase and Temple for your generous support and for attending tonight's event. Please stand so that we may recognize you.
All of us?
Yes, all of you. So at this time, I want to start by briefly introducing tonight's panelists using very abbreviated titles in the interest of time. And then before we get started on our discussion, I'll take a few minutes to tell you a little bit about the Damon Runyon Cancer Research Foundation, and then we'll turn it over to Dr. Uzzo, who will speak to you about the Fox Chase Cancer Center. So with me this evening, we have Dr. Jonathan Chernoff, who is the Cancer Center Director of the Fox Chase Cancer Center. Dr. Bill Kaelin, Professor of Medicine at Dana-Farber and Harvard Medical School, who, as I mentioned, serves on the Damon Runyon Board of Directors and is Vice Chair of Scientific Programs. And he is also the 2019 recipient of the Nobel Prize in Physiology or Medicine. Next, we have Jennifer Kalish, who is an associate professor at Children's Hospital of Philadelphia and the University of Pennsylvania, and is a current Damon Runyon clinical investigator. And Dr. Robert Uzzo, President and CEO of the Fox Chase Cancer Center. Thank you all for joining us and participating this evening. So the Damon Runyon Cancer Research Foundation was founded in 1946, and for over 77 years, our mission has had a singular focus. We are focused on funding brave and bold. We support young scientists who are pursuing high-risk, high-reward cancer research, and their work focuses on all cancers. We don't focus on any particular cancer type because we know that an understanding of the fundamental mechanisms that change in cells that can give rise to disease is incredibly important for understanding cancer, how it develops, and developing better treatments and diagnoses for the diseases. We fund basic research through to work that is much more translational and clinical. And so that means that we fund scientists who may be basic researchers with PhDs through to those that are physician scientists trained in both the laboratory as well as in medicine and are able to bridge the gap between the clinic and the laboratory. Damon Runyon scientists over the years have contributed to each new advance in cancer treatment over these decades, from the very first use of chemotherapy to immunotherapy such as checkpoint inhibitors that you may have heard of, like Keytruda and Yervoy, and the development of CAR T cells. They've also contributed to development of targeted therapies like Herceptin, which is one of the first targeted therapies that has been used to save lives of women with breast cancer. Genomics for better precision and personalized medicine, development of the HPV vaccine for cervical and head and neck cancers, and development more recently of mRNA vaccines for pancreatic and other cancers. The list goes on, and so we are very proud to have been able to support this amazing research that's led to incredible advances and countless lives saved. And so our scientists' work continues. 13 Damon Runyon scientists have received the Nobel Prize and numerous other international accolades. And since 1946, we have funded over 4,000 scientists with an investment of nearly $450 million. 100% of everything that Damon Runyon supports and all donations that come to the foundation directly fund cancer research, not overhead. And so that's something that is incredibly important to us to ensure that we are able to provide scientists with the absolute most, the resources that they need so that they can pursue innovative ideas and have the best chance of making breakthroughs. Some of our administrative costs are paid for by our Broadway ticket service, and that takes advantage of longstanding relationships with Broadway theaters. And so I like to include that because it is a nice way of really incorporating our history. Damon Runyon, our namesake, was a writer and wrote many short stories that Guys and Dolls, for example, is based upon and is quite iconic. And so if you come to New York, please call us, and you can buy premium seats to Broadway shows while also supporting cancer research. So please think of us next time you come. So with that, I am glad to turn it over to Dr. Uzzo, who will introduce himself and speak a little bit about the Fox Chase Cancer Center.
Well, thank you. Thank you very much. It's indeed a great honor and a privilege to be up here with my esteemed colleagues and with all of you, very accomplished people that really have given great support to bring comfort and aid to people suffering from cancer. And a special thanks to David and Sandy for really always being so supportive of both organizations and trying to really bring a different way of thinking about this dreaded disease. My name is Robert Uzzo, as was mentioned. I'm the president and CEO of Fox Chase. I'm a urologist by training. I grew up in New York, the son of an immigrant, went to Cornell Medical College, residency both there and at Memorial Sloan Kettering before going to the Cleveland Clinic and then coming to Fox Chase, where I've been for about 20 years. And I want to tell you a little bit about Fox Chase. I think most people here know about us, but we're an institution with a very storied history. 1904 is the American Oncological Hospital, and I carry around on my cell phone words from their charter, but I'm going to go old school today and read it off an index card because I want you to hear what they said back in 1904. It was arguably the first cancer hospital dedicated solely to the treatment of cancer, and it said, "This is a hospital for the study of the cause, treatment, and prevention of cancer and other tumors, and the dissemination of knowledge of the subjects for the treatment and care of persons affected with cancer. The benefit shall be administered without regard to race, creed, or color." And that's what they said over 130 years ago about the cancer problem. And to this day, if I get on an elevator and someone asks me what we do at Fox Chase, I say, we discover and we deliver. We discover new ways to deliver better care. And through our history, that's what we did. So in 1904, that was the American Oncological Hospital. In 1927, the Institute for Cancer Research came on board because people realized that we just didn't know enough about cancer. We knew it caused a lot of pain and suffering, but we didn't know enough about it. And the answer was research. And so from that time forward, that's what Fox Chase's mission has been. And it's been storied by great strides and advances. In the 1940s, Hugh Creech first used estramustine. We called it the ICR chemical for the treatment of cancer, one of the first chemotherapeutics. In the 1950s, David Hungerford looked under a microscope that sits in our hallway and discovered the Philadelphia chromosome, one of the first bases for understanding the translocation, the crossing over of genetic material between chromosomes 9 and 22, causing the fusion of two genes called BCR-ABL, which cause chronic myelogenous leukemia. And the list goes on. From Burke Blumberg discovering the hepatitis virus and the association with liver cancer, for which he won a Nobel Prize. And the most recently in 2004, Ernie Rose, for understanding how cells deal with proteins that get degraded. And we've got so many more. The two-hit hypothesis with Al-Knudsen, which is the basis for so many other cancers, like kidney cancer. And so it's really with great pride that I sit here with these wonderful and esteemed colleagues to help answer your questions and perhaps give you some insights about what people are facing when they have that question, when they hear that word, cancer, right? 1 in 2 people will hear it, 1 in 4 will die from it. And that's what we're here about. So thank you very much.
Thank you so much. Bill, would you like to go next and introduce yourself and tell us a little bit about your work as
Great.
well.
Well, first of all, likewise, I want to thank David and Sandy for making this evening possible, and I want to thank all of you for your precious time that you're sharing with us this evening. So my father was first-generation college. I grew up in the suburbs of New York, and eventually I went to Duke and Johns Hopkins and Harvard for my clinical medical training, and I went to Boston in 1987, determined to be a clinician, but I had the very good fortune in the late 80s of meeting a brilliant physician scientist named David Livingstone, and David really was the one who taught me how to become a scientist, and it was very clear that the only way we were going to cure this disease was to understand it much better. So because of that chance meeting with David Livingstone and the impact that he had on my life, eventually I had my own laboratory, and as you heard, we won the Nobel Prize in 2019 in medicine for discovering how the cells and tissues in your body sense and respond to changes in oxygen, which has implications not only for cancer, but for many other diseases as well, such as heart attack and stroke. And it turned out once we understood this circuit, or the rules, so to speak, we could see how cancers were sort of hijacking the system so that they would get oxygen. And so that enabled us, working with our colleagues in the pharmaceutical industry, to help develop new drugs to prevent the oxygen in the blood from going to the tumors. So I've been involved in one way or the other with the Damon Runyon for about 20 years now. I think first as one of the reviewers who decided which young scientists we were going to fund. Later I chaired those committees, and then as you heard, I eventually became the chair of the scientific committee for the board of trustees. And I will tell you, you could probably guess this, but if you win the Nobel Prize, you have to very quickly decide how you're going to budget your time, because you have to be very protective of your time. But I feel very, very, very strongly about the Damon Runyon Cancer Research Foundation. I see the work it's done and is doing. It turns out my wife was a very prominent breast cancer surgeon in Boston, who ironically diagnosed herself with breast cancer. She then was a survivor of breast cancer, but eventually she, in an unrelated set of circumstances, found herself with a glioblastoma, which is a highly lethal form of brain cancer, and she died in 2015. And I'm determined that for our children and our grandchildren, we're going to get there. And we're going to finally eradicate this terrible disease. And one of the best ways to do that is to invest in the best and brightest young people, which is really the mission of the Damon Runyon Cancer Research Foundation, as you just heard. So thank you again for being here.
Thank you, Bill. Dr. Chernoff.
Sure. So I also want to thank the Marshalls for all their support for the Runyon and for Fox Chase, and also I consider them personal friends. I'm grateful for what they're doing tonight, and all of you for coming. So my dad was a doctor, and probably one of the last of the general practitioners, a guy who did house calls in New Haven, Connecticut. He was one of three at that time that still kind of took care of all the patients in New Haven, which is a city of 150,000 people. And what I remember most from those days, and definitely influenced all his children, of his 6 kids, 4 of us became doctors, was the relationship he had with patients. I mean, it was just amazing back in the day. I mean, of course, my dad was a great guy, but that model of going to people's homes gave people a totally different feeling about their relationship with their physician. And there was no talk about clients and healthcare providers. It was doctors and patients. And they really, I mean, I think they loved him, but he also loved them. And it was just a relationship I wish that we could maintain in this century. It just doesn't seem to be any going back. But it was definitely made me want to become a doctor. I wanted to be just like my dad, and so did my brother, my other brother, and my sister. And I don't know if we became like him, but at least we became doctors. Now, I was bitten by the research bug pretty early on, and for the longest time, even arguably till today, I couldn't decide what I really wanted to do. I liked science, I liked medicine, and I tried to do both, and so I ended up doing an MD PhD, and these days, the only patients I have 4 legs and a tail. But I'm still board certified. You wouldn't want to see me as your doctor anymore. But and frankly, most of the mice, I'm giving them cancer, not taking away. But I trained in an area called signal transduction. I trained with Ray Erickson, who was a guy who found one of the first of these oncogenes called SARC. And it was an era of sort of the ultimate reductionism in cancer. An important era in that they were beginning to discover the drivers of cancer, these genes that were mutated in cancer, and SARC was one of them, and RAS, and a lot of other ones. And I spent, and I still spend, a good part of my career studying these sort of drivers. And as you know, the pharmaceutical industry has been quite adept at making drugs against some of these drivers. And I thought, and I think many people thought, we were about to enter the golden era of cancer therapy because we knew the drivers, we now had drugs, and it's been a bit of a tarnished golden era in some ways. And we can get into this later with some of the questions, in that perhaps we should have expected, but didn't at the time, that it would rapidly engender resistance. And part of all of that, and again, I'm not going to take all the time right now with this question, but was due to the way we were studying things, this reductionist approach. It didn't take into account systems, that cancer cells don't exist in isolation. They're part of a microenvironment. There's an immune system, there's a microbiome, there's a nervous system, all of which affect how you respond. So I think that was one of the issues that we've learned in the last while, which I am now focused on, that I wasn't doing 20 years ago. Thank you.
Thank you. Jen?
Thank you, and thank you to you for inviting me here and for being here to hear the story, and most importantly, thank you to the Damon Runyon for funding my research over the past few years. My cancer story begins actually in middle school, where I had a good friend who was diagnosed with brain cancer, and there was very limited treatments at the time. And what I learned going through that process with her was that you had to think outside the box. So that took me to college, where I studied chemistry and physics, knowing I'd get back to medicine eventually, and then to graduate school studying genetics. And I'm actually not an oncologist, I'm a geneticist by training. And as my training continued, I ended up at CHOP for residency in pediatrics and genetics, and I met another little girl, about the same age as my friend actually, who actually had another kind of cancer, a Wilms tumor. And in that case, thinking about the kinds of kidney cancer and the kinds of things we need to think about beyond just the cancer is the genetics and the other approaches to it. And that led me to thinking about research and how I can apply the training that I've had as a geneticist to cancer. So the work that I do now is focused on something called Beckwith-Wiedemann Syndrome, which is a cancer predisposition syndrome, something that predisposes children to cancer, but not all children develop cancer. And when I brought this proposal to the Damon Runyon Foundation, it actually met with what they call the handshake rule, where they want a translational research approach that goes from the patient to the science and back. And being able to use the expertise in genetics that I had developed and this patient population, we were able to start studying all these factors that actually occur in many different types of cancer, kidney cancer, liver cancer, even breast cancer sometimes, but having a patient population that has all of these changes that we can develop cell lines and models to study. And that's one of the things that over the past 5 years has really been able to drive this forward, because doing this translational research, there's not a lot of funding mechanisms out there that will allow you to really bridge that entire gap as you're trying to build a research program. And that's something that has really been an amazing thing that the Damon Runyon Foundation has provided me as I've developed both as a physician and scientist and a researcher in this area.
Thank you. So as we've all, I think each one of us has mentioned some of the amazing progress that has come about in cancer treatment and research. And so I wondered if one of you could speak to some of the more significant recent advancements in the field and what new directions you're most excited about right now.
Well, I'll start. I consider sort of the Big Bang in all of molecular medicine, including cancer medicine, being the year 2000, where some of you will remember we had the first draft of what's the so-called human genome, or the DNA sequence of a human being, and in particular, the correct DNA sequence of the approximately 20,000 genes we all have. And so you can think of that as being sort of the parts list finally. Cancer, in the end of the day, in almost every case, is caused by a rogue cell where a handful of genes have become altered, scientists say mutated. But before the year 2000, again, we didn't even have the full complement of what should the normal gene look like, let alone can we recognize that this gene has an alteration or a mutation. So for the past 20 years, now that we have the parts list, we're increasingly, with modern tools, able to look at any cancer, figure out specifically which genes have been altered, and then increasingly to match those alterations with so-called precision drugs that target that abnormality. Now, Jonathan is completely correct that in many cases, when you use these new precision drugs, you get a dramatic response followed quickly by resistance. But what we've learned from other diseases like AIDS and even before that tuberculosis, the trick is to combine several drugs that have very distinct mechanisms of action so that the chance that any one cancer cell is going to be resistant to all three becomes almost zero. And so I think we're in a transitional period where we increasingly have to use drugs in combinations. And then the other thing, as Jonathan correctly points out, is it's one thing to focus on the cancer cell itself, but the cancer cell is talking to the host, and the host is talking, which in our case is the patient, and the patient's tissues and cells are talking to the cancer. But now we're learning those rules too. We're learning how to listen in on that conversation. So for example, as was mentioned, we now have new drugs that rev up the immune system, so the immune system will see the cancer as foreign and destroy it. But to do that, we had to first understand all the ways that the cancer cells were cleverly and sinisterly cloaking themselves so that the immune system didn't even see the problem. But now we're starting to remove those cloaks, and now the immune system's going in. So I think we're in a very exciting period, but we do have to get to combinations, and it is going to take a multi-pronged approach.
Well, some of you know that Bill is one of the masters of synthetic lethality, which is another way of talking about targeting multiple things that would be lethal in a tumor if you hit more nodes than one that are not so lethal in a normal cell. And that has become all the rage. I think there's not a week that goes by that there's not another combination that is being touted. I think another advance that wasn't really appreciated 20 years ago, because it was all about genetic lesions, was epigenetics, a sort of upper layer of gene regulation. We still don't really have a great handle on epigenetic regulation, which is generally involves either methylation or acetylation of DNA or histones that DNA wraps around. But that is integral to how genes are expressed. And in most cancers, arguably all cancers, epigenetic regulation is altered in some way. And what's new in this is not just an appreciation of the importance of epigenetics, but that there are actually targetable drugs there too. That it's not always the tumor oncogenes per se, but it may be things that are more subtly affecting gene regulation. So that's a whole new vista, and there's many companies and many drugs out there that target this epigenome.
And I think as part of that, I didn't mention before, but the syndrome I study is an epigenetic syndrome, and there's actually a number of known genetic syndromes now that we're understanding more about that we can actually use as models to try and do some of this targeting for the epigenetic mechanisms, because they're things that already exist and are compatible with life, but we can actually find ways to understand them and modify them.
Well, I'm the least scientific of the members up here. I did spend a couple of years in the lab, 2 years, but I certainly don't spend my time there. So I get most of my inspiration from the questions patients ask. And one of the most fundamental questions I ask is, what caused this? And so for years, the answer was just so unsatisfying. You just didn't know. And so over the course of the last decade or so, there have been all these studies about cancer evolution and the evolution of a cancer cell. And to me, these have been really foundationally, they change the way I think about this disease. Really, it's an ecosystem, as John said. It's not a single cell. It's a cell that exists in an organism, in a microenvironment, that dovetails with the immune system and with all other aspects of that person. And so that evolution occurs sometimes in a very random fashion. We call it stochastic. And so sometimes cancer is just a replicative error. And this really, as you start to try to explain this to people in ways that they can understand, because everybody wants the control that goes along with understanding, replicative errors are as if I gave you a copy of a book and I asked you to Xerox it 1,000 times. And I asked everyone who received that copy to Xerox it 1,000 times. And to do that 1,000 times times 1,000 times. Well, suddenly the P's start to look like O's, and the O's start to look like C's, right? And suddenly it becomes unreadable. And even though you've got proofreading mechanisms in your cell, those mechanisms over time age, and your immune system ages. We call that immune senescence. And so what happened when I was young is we would be describing what we see. We see a very robust inflammatory change within a cancer tumor, within a tumor. But we don't know why that inflammation is not doing anything. Well, the immune system's aging. And we started to think of ways that we can sort of change that calculation. Can we give cytokines or drugs to boost the immune system? Can we take out those lymphocytes within a tumor cell, put them in a petri dish, boost them up, and give them back to you? Or can we figure out why the cancer cells have evolved in a way that they can hide themselves from the inflammatory cells. And that's where we are right now. And so it's all about that ecosystem. Cancer is an ecosystem, a very unique ecosystem within each one of us. And if you start to understand it that way, you can understand the concept of personalized medicine as well.
Would you like to speak more about
Well,
personalized medicine?
You
opened it.
Dovetails us right to there, right? So,
Exactly.
so personalized medicine, right? So, typically, back when I was training, if you had bladder cancer, you got this regimen. If you had kidney cancer, you got that regimen. And of course, we know that it didn't work equally well in everybody. Some people had results. Sometimes they were great. Often they were marginal or none. And so the concept of personalized medicine is ultimately understanding who is most likely to respond to treatment A, B, or C, or combinations in certain sequences. And to do that, you need to understand the unique ecosystem within that person. And so that comes to the notion of genomics and proteomics and all sorts of these unique, very sort of bioinformatic intense, analyses that we're starting to do and understand to predict response.
Thank you. And Jen, I was wondering if you might want to add to with your background as a geneticist, how does this advanced understanding of genetics and how it drives disease affect the way that you can treat your patients today?
No, I think it's a great question. I think it's a matter of being able to take the tumor and take blood from a patient, or even a tumor in the normal tissue next to it, and sequence it. And basically look at all of the genomics that you were just talking about, look for the genetic changes, and use that in a evidence-driven way, because we know that the genetic change over here means that this drug will work for this patient and this drug won't. And I think because technology to get that sequencing is much more rapid now, and we have access to the tissues and the bioinformatics, the way to interpret that data, we can actually make it a much more tailored approach where it's not just this drug is for you because you have kidney cancer, this drug with this genetic change means that this kidney cancer will be treated, whereas this one may not. So a much more tailored way. And then there's also the advent of immune therapy approaches too, based on some of those genetic changes, and thinking about CAR T-cell and things like that, where you can really derive basically treatments that are specifically for that patient based on their own cells, using some of these genetic factors.
One of the things that I found most interesting recently was this study by a group in London called TraceRx, and it gets to this concept of the ecosystem, right? So what they did is they identified people's kidney tumors in this case, and they've done it in colon and others, and they kind of backdated all of those various genetic events that may have occurred as the clone evolved. And they backdated it as far as what they thought the original defect was. And what they identified, and I think pretty convincingly so, I'd be interested in Dr. Kaelin's thoughts here, but the original defect, the original cell that became malignant in a 65-year-old really occurred with a mechanism called chromatopsis at sometime when that person was between the ages of 15 and 20. Now think about that for a minute. I just said this is an ecosystem, and I just said that ecosystem, has profound implications for how you treat that unique person. But think about what I just said. Can you prevent cancer if the very basis of that ecosystem starts somewhere between the first and second decade of life? And then we start talking about cancer prevention and cancer interception. And that sort of leads us to many, many different sort of, in some ways, medical hypotheses to be tested, but also some moral implications that might have, right? I don't know, Dr. Kaelin, if you can comment on your thoughts?
Well, you're absolutely right. I think I like to use the analogy of cancer being sort of like tumblers on a lock, where, say, 3 or 4 or 5 tumblers had to fall into place before the lock would open, but in this case it would be the cell going from completely normal to completely cancerous. So the good news or bad news, depending on your point of view, is it's very clear with modern molecular techniques that, first of all, what Robert said is true, that most cancers, by the time they're clinically overt, we can see them on an X-ray, for example, but if you really use modern molecular techniques, that initial initiating event probably occurred years and more likely decades earlier. But perhaps more importantly, as we think more about how to use very powerful screening tests to look for cancer in the general population, every single one of you has some cells in your body right now where the first 2 or 3 or 4 tumblers has fallen. Certainly the first couple tumblers have fallen. But thankfully, it's still a numbers game, and hopefully the final tumblers never fall. But as we get more and more and more powerful techniques, there's no question that we'll be able to do, for example, in time, a blood test that would say, somewhere in your body there's a cell someplace where all the tumblers did fall. We can't see it yet, but we're worried about it. Well, you can imagine the technology getting ahead of what could we do practically, and if we don't get that right, we'll do a lot of harm, right? Because we'll scare the bejesus out of people, but even the doctors may not know exactly what we're supposed to do with this information. So this is an area of intense research that at the time the diagnostic tests are getting more and more powerful, the prognostic tests get more powerful, and our ability to say, nonetheless, we don't have to worry about this abnormal blood test versus we better think about this differently, and maybe we even need to do some form of chemo prevention.
It is a kind of a scary problem. You think about, the so-called biomarkers we have for cancer, like for men, maybe PSA for prostate cancer. Even after, how many years has that test been out? 20, 30, 40 years? We still don't really know what to do with it. It's still a much debated topic about what the value of screening general population with that, which is a relatively simple thing. These molecular tests, and I'm talking about, I'm a person who actually took the Gallery test. I was curious, I'm affluent, I'm knowledgeable about medicine, I want to see. And you get back either something grim, like you've got some marker that looks bad, or in my case, it looked good. But how would you ever apply that, as Bill says, to the general public? We don't even know what to do with that information. You have a RAS mutation. That sounds awful. It probably is awful at some level, but it may not be clinically relevant at all. And we see clonal hematopoiesis, where your blood changes how many sort of stem cells you have. That happens. It happens as you age. It's not necessarily a terrible thing or a sign of anything awful, but it can be. So we just don't know enough to deal with these at the moment. But those tests are out there now.
Yeah. Since this sounds a little bit almost like a downer, I think this is good news, but another observation that goes back to the middle of the last century is if you look at men age 80 who went to autopsy for any of a number of reasons, they got hit by a bus. Yeah. Half of them, if you look really closely at their prostates, had early prostate cancer. It was never clinically manifest, and probably in some cases never would have been clinically manifest, but if a clever pathologist looks hard enough under the microscope, they can find it. Well, the same thing is happening in all our organs. There's no reason to think it's peculiar to the prostate. So the good news is a lot of these cancers, they hit a dead end and they're never going to be a problem, but that's what we have to figure out. Which ones have hit a dead end, they never would have been a clinical problem, versus this is the rare type of cancer that would have gone all the way to really harm you if given a few more years or decades.
And I think one other thing to add to that, I think pediatric cancers gives you the other end of that spectrum, where you can learn a lot from what's happening early on. And as we try and understand more about pediatric disease in general, more and more children are being sequenced and looking at the entire DNA sequence, whether they have cancer or not. So we're going to have more of that information of what's the normal baseline that may not lead to cancer. So we can look at things even before that 15 to 20-year-olds, but knowing even from well before that eventually. And that's the power of, in 20-some years, going from looking at just parts of the DNA to looking at everything.
I mean, to underscore the point, if you get a CAT scan and you're looking for cancer and you've got a 1 centimeter nodule, that's about the limits of detection where you say that's probably abnormal, 1 cubic centimeter. It's about 1 trillion cells. 1 trillion cells, and they are very diverse. And within that 1 trillion cells, there is, again, this dynamic ecosystem, a survival of the fittest, if you will. Many of those cells will never do anything, maybe all of them. And you'll, as Dr. Kaelin says, you'll go on to die of other causes. And yet, some of those cells can sort of acquire the ability to do bad things. And distinguishing those two is really the art of the biomarker. And as Dr. Chernoff said, the art of that biomarker is now sort of coming to age with circulating tumor cells and circulating tumor DNA, the ability to measure in a simple blood test, which frankly, I'm always surprised. There's lots of patients that really don't live in these worlds, but they're affected by it, that think that, there is a simple blood test that already exists, and they ask for it frequently, but no such thing yet exists. But if you think about the layers and layers of complexity, and as Dr. Kaelin said, there's lots of good news here, but there's also sort of, it is foundational and building on itself to understand that this evolution and our ability to intercept that evolution really sort of are colliding right now in our ability to predict the next event.
So you've all touched on the fact that we are now able to collect vast amounts of data, and we're improving our screening. And one of the questions that comes up is how are new technologies like machine learning and artificial intelligence going to impact our ability to take all of this data and actually use it to better understand disease and actually improve human health.
It's a tough one.
Well, I think clinically, Rob, you might have the
Well,
best
I'll take a
insight
crack
on
at it.
this.
I use ChatGPT. I like to talk to. I use ChatGPT. ChatGPT right now is very good at making very binary types of decisions. What's the infection rate after a hip prosthesis, for example? But it's not very good at judgment, right? Because it hasn't acquired judgment. Will it get there? Probably. But the complexities of cancer care and the trade-offs very, very much depend on the conversation, right? I mean, so it's risk tolerance. I have a lot of conversations with patients about their risk tolerance. In fact, I had that conversation with my father. My father died of prostate cancer. And, a couple of months before he passed away, I asked him about that risk. Would have you liked to know about this 35 or 40 years ago? Would you have lived with the consequences of treatment at that time to avoid death from prostate cancer 3 or 4 decades later? And it was very unique what his answer was. His answer was to me, no, I had a good life and I would not have wanted to endure those complications. That was his decision, at least. And I thought that was to me it was very eye-opening because it was at a very, vulnerable time, right? He was dying of the disease. And I think that AI won't be able to sort of give you that level of judgment. It can give you some information with which to base those conversations off, but I don't think it will be able to substitute for the discussions that occur behind a clinic door.
Yeah, I have a little bit different take on this. So, I mentioned that my dad was a GP. He was also, he had a specialty. He was EKG guy, and he read every cardiogram that Yale New Haven Hospital back in the day. And, you think about artificial intelligence, its first application of the type I think that we're thinking about now was really in his specialty. He wrote books on EKGs, but already by the 70s, there were primitive computer programs that were reading cardiograms because they're relatively simple two-dimensional squiggles that you can measure rates and do quite a lot. And so he was maybe the last of the human readers only of EKGs, but even in his time, and he died in the 70s when he was quite young, but it was already beginning to approve pre-read EKGs that these primitive computers were doing. And now, of course, all the hospital EKGs, the first look is by the computer. It tells you what it thinks. You still have somebody signing off. But that same general idea is present in many other parts of medicine now. With advanced, now we're in the era of, ChatGPT and other things, reading a pathology slide or a X-ray type study. I have no doubt they're already doing it, but the first pass will be machine, and then hopefully you still need a human somewhere in there, but maybe not. And they've already tried it in pathology. It's already being used for pap smears and things like that. It can breast mammograms and such. The limit on this, I don't know. It's not my specialty, but I can see a deep inroad being made in those diagnostic tests.
Well, I would say 2 things. One, as a Yale medical student, we did all have to learn to read the EKGs, thanks to your father. And also,
you might have used his book because
we did use their book. But in talking about what you mentioned before about the size on a CT in which you can see something, one of the advantages for AI is that you can take that size down farther and look for patterns in a way that the human eye can't necessarily pick up. So I think there is, and combining that with genomic information, proteomic information, I think there's going to be ways to develop algorithms that maybe we have to determine whether or not they make sense and we should take them back to our patient. So the human component is still there, but there's going to be connections made that we may not be able to make at this moment in time. So that's going to be one of the huge advantages we're going to see as we start figuring out how to apply the technology.
Yeah, I think that's true. I think ultimately it'll make us better, just like it makes pilots better, but you still got to land the plane, and I'm not sure we're there yet.
So I think unquestionably AI is here to stay. And as Young pointed out, we need AI because of the enormously large data sets that are now being generated, in part because of something we've talked about today. So your electrical device here was made possible by the precipitous fall and the cost of transistors and integrated circuits. But the only thing that actually has fallen faster is the cost of sequencing genomes. So the first genome cost several $billion, and we all paid for it, by the way, with our tax dollars. But now it costs less than $1,000 to sequence a genome. And so now it's just a matter of fact that people sequence cancers, and now we even have the technology to go within a given cancer and start sequencing the genomes of the individual cells within that cancer. So this starts to get big data. And then there's imaging we talked about. But I just get two cautionary tales, because I think we're in a transitional period where the hype sometimes gets blown up. So there was an early study where the computer was shown chest x-rays that either had lung cancer or a benign lesion. And it turned out with AI, the computer was 100% accurate, saying this is a lung cancer, this is benign, this is benign, this is lung cancer. So you get pretty excited and you see the thing go along the bottom of your TV, headline news maybe, AI can outperform radiologists. When they look behind the curtain, what the computer was doing is the computer figured out that the cancer x-rays have been done at a tertiary referral center with a really fancy x-ray machine. The normals had been at a primary care facility with a less sophisticated x-ray machine. So in the end, it was what we call a hidden confounder. It was of no interest whatsoever. I mean, it looked great, but that's not what we're trying to do here. I could give you a number of these, but I want to make sure we have time for your questions. So.
So thanks, Bill, for sharing that. We've talked a lot about the fact that there are a number of new treatments for patients now, immunotherapy, targeted therapies, but the need to create and understand combinations that will work better for each individual patient. So what is the role of clinical trials, and why are they so important in moving us forward?
That's for, you want to say that? That's
Sure,
yeah, I think that's the end of the table,
question. Yeah. Well, so I've been involved in clinical trials for many, many years, and I always start by thanking the patients. I mean, it is not easy to be a patient and understand the concept that someone's going to flip a coin or a computer doesn't randomly assign me to treatment A versus treatment B. That takes a great deal of faith and confidence. And frankly, as you know, Fox Chase is part of Temple Health. And one of the greatest assets of that is we have the ability to go into cities where there is, the city of Philadelphia, where there is a great, disparity in outcomes. And we have to get people to trust the concept of clinical trials. So, that's a little bit parenthetic. But ultimately, they say that progress is made very slowly and then all at once. And the all at once part is a result of clinical trials. And clinical trials often take, a decade before some drug comes out of the lab and gets approved by the FDA. And I think we all know sort of the roadblocks and the obstacles it takes to get there. But clinical trials really have revolutionized the care. I'll use kidney cancer as another example. When I was a resident, basically, if you had metastatic kidney cancer, you had 10 months to live. That was only 20 years ago. I'm halfway through my career, and now the average survival is about 5 years. So that's a 6x improvement over half a career. I think you haven't seen that much progress since, I'd argue, since the advent of antibiotics, perhaps. And clinical trials did that. I don't know if anybody wants to add to that.
Well, the only thing I'll say is that in both science and clinical medicine, there are many, many examples where you encounter something and you say, it's intuitively obvious, this is going to work, or it's going to be this way. And then you subject it to a formal experiment, in this case, a formal clinical trial, and it turns out your assumptions were wrong. So the only way is the scientific method, clinical trials, test your hypotheses, and that's the way you advance.
Mm-hmm. Yeah, and I think in this field, in cancer, it's probably the one that needs clinical trials the most, because, again, I don't want to be like the downer here tonight, but our treatments are just not good enough. I mean, we have a lot of things we've learned over the last century, really, and certainly accelerating over the last 20 years, but a lot of people get cancer, still die of cancer. It's just unacceptable. And there's hardly any cancers where we're so good at it that we couldn't do better. And so that's where the clinical trials come in, because we don't have great treatments. If we did, we wouldn't need any trial. We just do what we're doing. But that's just not generally the case for most cancers. So it's super important. It is difficult to understand, as Dr. Uzzo mentioned. Patients come in, they're confused enough as it is, and they need our help. And sometimes our help is putting them on a trial because it might be better than the standard of care.
I'll just take you through a short conversation that people have, like in the clinics. So in my field, you've removed the tumor surgically, and the patient comes back in 2 weeks, and they want to know the pathology, and you have a conversation that goes something like this. The operation went very smoothly. There were no complications. I'm happy we were able to take you through this safely. Your pathology result came back, and the predictions are that you've got a 30% chance of recurring in the next 5 years. And so the logical question that they ask is, well, what can I do to lower that risk? And then often, the answer is, we simply don't know. In this particular tumor type, there is nothing that's approved, and yet there's a clinical trial. And they say, okay, well, tell me about that. And you say, well, the trial looks like this, and, you get randomized, arm A and arm B. And arm B is often a placebo. And so they want arm A because they want to do something, right? They always want to be active. Of course they do. And their inference is that somehow the active arm is always going to be better. And sometimes it's not. As Dr. Kaelin said, sometimes actually it does harm. And so then the question is, okay, well, if I do take the drug, what are the side effects? And you say, well, there's a 10% chance of this and a 20% chance of that and 30% chance of this. And so then they sort of do all this mental math. And in the end, with informed consent, they get very confused and they look at you and they say, what would you do if I was your father? And that's always the question. That's always the question every time. And that's, and then you bring the human aspect of it and you give them your best shot at it. But they take a chance on you and you have great respect for that.
Yeah. But I think it's important to point out, I'm usually a lumper rather than a splitter, but since you were a lumper now, I'm going to be the splitter. I think it really depends on what kind of cancer you're talking about. I think this is really important. I mean, there are some cancers that were absolute death sentences 10 or 20 years ago where we made remarkable progress, where either people have been cured, there's clearly a subset of people with metastatic melanoma, for example, who are now cured. You mentioned patients with kidney cancer, 5 years. Anecdotally, I know some people over a decade, chronic myelogenous leukemia, multiple myeloma. I could go on and on, but the point is, once you start making that kind of real transformative progress in a couple cancers, some of the rules start emerging. And so those rules are emerging. So I think there is a playbook that we can start to apply to the cancers that have been more difficult.
And so are there ways that we can help cancer patients to become more empowered in their journey? Obviously, part of this is education, and I think there's a role for advocacy as well from scientists, researchers, people in this room who are really passionate about the cause. So where do you see that there are places that we can do better?
Well, one of the NCI occasionally gives us new mandates that we're not always thrilled with, but one of the ones that they did, an emphasis there that I think was long overdue at cancer centers like ours and others was what we call now community outreach and education. It's abbreviated COE, you talk about it in that way. But it's something we've always did, but have never done well enough. And it's become almost on par or maybe equal to the science that we're doing is how we disseminate what we're doing into the community and vice versa. What are the concerns of the community? What cancers do we even affect our community more than others? And the so-called catchment area, you'll call it. And in Philadelphia, that looks very different than if you were out in Oklahoma or somewhere else. So I think the cancer centers in general, and certainly ours, is much more attuned to what's going on in the community. And we have programs, we call them community ambassadors or liaisons, people who actually come to our science meetings from the community or attend our clinical trial meetings and may have different suggestions. Sometimes you get, why aren't you doing any trials on pain or nausea? They tend not to be in those areas, but those are a great concern to patients. So having community representatives inside, if you will, and us going outside, if you will, is of great value and I think changes the calculus of what we're doing.
I think there's also a role for partnership with foundations and patient and family advocacy groups, because there are a lot of family foundations formed around particular types of cancer, and bringing them in as trials are going on. A lot of times they're actually even part of the application for the trials to help spread the education that way, and also in designing the trials.
I think historically, hospitals and to some degree physicians have treated cancer, as an episode of care. It starts with the diagnosis, it ends with the treatment. And so, we could be, I think it would be a legitimate criticism that we're guilty of sort of seeing it like this episode as opposed to this journey. And so what cancer centers like ours try to do, and the Conference of Cancer Centers across the country, is they look in front of that journey, the screening, the interception, the family risk, and they look behind the journey. Increasingly, it's things like survivorship. And the NCCN, which is the National Comprehensive Cancer Network, of which we're a founding member, and they put out all these guidelines. They talk a lot about survivorship. So fatigue associated with survivorship, secondary malignancies. Sometimes the treatment we give you induce another cancer. They treat one cancer, they cause another cancer. Cardiac risk associated with chemotherapy, for example. I mean, there's a whole guideline on survivorship, as well as the mental health, the mental well-being, and all the sort of things that go on, even beyond 5 years of cancer treatment. So I would say one of the things that, cancer centers, increasingly are focused on is the bookends of the episode of cancer care. And that's really where I think, a lot of the difference is made and where patients really become far more empowered because they feel a great sense of loss of control. Every time I walk into a room and I see a patient having had surgery, I think to myself as they're laying in the bed and we tower over them, that they really have, this is so foreign to them. And where they get the control back, I think, is hopefully how you treat them in those moments and sort of on the bookend.
I have this one small anecdote that your story reminds me of. We recently hosted 4 physicians from Ukraine that come to Fox Chase for 2 weeks. And they wanted to learn about, they'd heard this American style of treating patients through service lines. They didn't want to learn more about that. So we had them here, and they ended up giving a talk about their experiences doing cancer care currently in Ukraine. Is pretty tough. But one of the things came up with clinical trials, they saw how much effort and time we put into this. So, the way you interact with patients here in the U.S. is very different than there in Ukraine, in that you actually let the patients ask questions and you answer them. Apparently, and I may be overstating it, but according to our guests, in Ukraine, generally, the doctor tells the patient what they're going to do, the patient says okay, and then it gets done. I think the American public has been educated to an extent, needs more still, but it's very different and in a healthy way. I think with that interaction with patients and doctors, each asking questions and not just accepting things as dogma is the way to go.
So I'd like to switch gears a little bit and talk about the fact that if we want to continue making progress and have further breakthroughs, then we really need to be able to invest in research and continue supporting these amazing scientists and physicians that are working on understanding the disease better and developing new treatments. So what are currently some of the more significant obstacles toward making progress?
Well, I think there are several. I mean, first of all, I've said I don't want someone going into science because of the money, but I don't want them not going into science because of the money. And there are opportunity costs that bright young people have, especially if they have a STEM background. For example, the Googles of the world will sometimes hire a undergraduate from a Stanford caliber place and pay them more than a typical biomedical PhD or postdoc. Gets paid. So, if you're a bright young kid and you're kind of STEM, you work on STEM and you're kind of quantitative and you got it all going on, you can, maybe door number 1 is Goldman Sachs and becoming a quant person, or maybe door number 2 is Google. And then we say, well, but you could get a PhD in biomedicine. That'll take about 5 years. And then, by the way, you probably need to do a postdoc to be competitive. So that's another 4 years. And by the way, you might be still living on ramen noodles. So I think we have to figure out, if we really value this, how we recruit and retain some of these bright young people. And my mentor David Livingston used to always say, Billy, this is our seed corn. If we don't get this part right, there's no future to pass on to the next generation. So we better nurture and support some of the best and brightest, which is, again, why I think the Damon Runyon plays such an important role in the entire biomedical ecosystem, because this is what we talk about all the time. How can we support these young people?
And I think by offering support at every step of that process, which is something that Damon Runyon does, not just in terms of the funding, but also I've had some of the members of the initial judging panel who reviewed my grant, I'm still in contact with as hurdles come up, or there's a new technology we want to apply. Do you know somebody who can help with this and form new collaborations? In fact, you just connected me with someone who's junior to me, who I'm going to be helping set up a registry for patients, because that's something that I've done as part of this work. So I think that continued support at key junctures during the training process is really important, because there's a lot of federal money out there, but the foundation money is really what helps fill in sort of the meat of a lot of projects, because because even just having an NIH grant is not enough to support even a small laboratory at this point, especially if you're doing this very complicated translational research.
You know, Bill and I are roughly the same age, same cohort, and so we came up at the same time. And you know, in the 90s, things were rather different than today. It wasn't always this completely life and death struggle for funding, which is what it feels like today. I mean, that was the era of, you will call it, the NIH doubling, and you know, times were pretty good for us scientists, and would that have stayed that way forever? But the last 10 years, certainly, and I don't know what's going to happen down the road, but it's just gotten so difficult that a lot of young people don't want to put up with it. And there's a lot of biotech out there, there's a lot of other opportunity, and people are taking that opportunity. So I do worry about the future in that way. It's, as you say, that seed corn. Guys like us, we've got a few years left, maybe, but these other people, if they turn, I mean, biotech's not so bad, but it's not discovery science, really, for the most part. We won't learn the things we need to learn to get rid of this disease.
I would just add that the medical industrial complex is a huge bureaucracy, and I say that lovingly as a leader of a bureaucracy. Frankly, I think that it used to be that margins on clinical medicine were very robust, and a lot of that flowed forward to sort of discovery research. At cancer centers like ours, that still happens. But frankly, you know, a 3% margin is tough to sort of support a large research endeavor, and most hospitals exist on that level of margin. So, of course, it's time and money as always, and of course, it's specimens. And I'd say that we're now growing the infrastructure to do that better. Most large cancer centers have a very large biosample repository. And, you know, and how you do that, and how you organize that, and the infrastructure to do that, so that you pair the urine and the sputum and the, you know, the tumor, you know, and the clinical outcomes data on that patient, so that you could give it to the scientists, and they know, you know, this is the enriched population I want to look at to get the most, you know, the most information from the science and the studies that I'm doing.
And I think just building on that, that's part of the partnership between foundations and institutions, because starting those biorepositories and maintaining them and continuing to add to them is a lot of work and a lot of personnel time to make sure you've got the right part of the right sample going to the right process. And that's something that's really important with this, with back and forth between, and it's infrastructure grants and things like that that foundations offer, and even some of the basic science that's part of it, and with large institutions like Fox Chase, like CHOP, that you can do that.
What do you think we can do better to recruit and retain more young people to pursue careers in research and in medicine?
Well, I don't think it's that complicated. I think it really relates to what we just said. I think, again, I think this is the best job in the world, being a scientist. But in the end, if you're a young person and you're raising a family, you think about the same thing everyone else thinks about. Am I going to have a stable source of income to support my family and take care of their needs? So I think we need stability. I think we need to sort of increase stipends a little bit to get them catch up a little bit. But I think at a minimum, I think there needs to be, you know, stability, that they know there's a career here for me and that I'm valued. And the other thing I would say is, it occurred to me after I did get that lovely phone call from Stockholm, you know, all the things I benefited from. You know, I remember going to my dad's office on Wall Street and watching the astronauts come back from Apollo. And I remember how, you know, during the space race in the Cold War, you know, the scientists and engineers, they were the good guys. They were, unfortunately, it was mostly good guys. There weren't enough good gals. But they were the heroes, and the language from all political corners was, we got to do this. And, you know, again, smart people hear language being used. And now, of course, we've seen it's gotten a lot more complicated. So I think we've got to get the messaging right, too, that we believe in science, we believe in research and generating new knowledge.
I think you got to start really early. We have really excellent programs in our Institute for Cancer Research for high school students. You know, you want them to be in lab. You want them to be exposed to the scientists. You want them to see sort of the interesting stuff that's going on. You see them when they look under a microscope. They've never actually seen a cancer cell, and sort of this light bulb moment that goes on. If you can get them early and get them interested, and then they'll have a life experience that'll want to lead them in that direction more often than not. I think that mentorship is you know, foundational.
Yeah, I would reinforce that too. You know, I'm nominally the director of Fox Chase Cancer Center. I say nominally because scientists, by definition, don't have bosses. That's why you do science. Nobody tells you what to do. Otherwise, you do something else. And so, it's one of the few jobs that you can have a dream or something at nighttime and then come in the next morning and execute on it and see if you were right or wrong. There's nothing like it. It's like a drug, but get that drug in early, because most people, if you ask them, do you want to have a boss? I think they'd say, no. I want to be my own boss. And you can actually do it and have a successful career and benefit mankind all at the same time. So if it weren't for the lousy funding problems and some academic politics that come up from time to time, it'd be like paradise.
And I think taking it out into the community, also into schools, and even hitting even before high school when it's too early for students to come into the lab, but just bringing it into the classroom and going out and speaking in the community and the schools so they can see this is what a scientist is, and explaining all of the things you just described, but just having it out there in a very accessible fashion for anybody.
We give these students their own experiment. They don't just come and watch, they actually do. Right, and most of the experiments are relatively simple and basic, but we actually get them to sort of design or help design the experiment and conduct the experiment, and that's really what gets their passions going.
Amazing, thank you. So we would like to reserve a little time for questions, and so we're happy to take questions from the audience, and I think we have microphones at the back of each of the stairwells, so please, I see one hand raised here. Let's start there, thank you.
First of all, I'd like to thank David and Sandy Marshall for hosting this event or putting it on. I really appreciate it. I have kind of a basic question as a patient. Chemotherapy, it feels like a very blunt instrument. You talk about targeted therapies or immunotherapy, but as a patient, it feels like chemo, is very much a blunt instrument. So my question is, for somebody that has like stage 4 colon cancer, how much of the chemo is preventative, because the cancer's like floating around your body and other potential dangerous areas, versus targeting to like where the cancer is? I'm just curious about that.
Well, I should start with a disclaimer, which is my medical license is no longer active, so this is not formal medical advice, and I hope you're doing well. I mentioned the year 2000 as sort of a watershed moment in the history of cancer medicine. So it's remarkable the drugs we did develop back in the end of the last century, but we were blind. We didn't know the genes that were altered. We didn't know how to, and so we didn't really have the parts list, and we didn't know which parts were abnormal in specific cancer. Now, for colon cancer, routinely, we have a pretty, we can go in and understand what genes are probably altered, have been altered, and are driving that cancer. Now, conventional chemotherapy was, when most people say conventional chemotherapy, that they were discovered, again, blind to the actual genetic causes of those cancers. Now, some of those drugs turn out to be very active, in some cases life-saving. For example, Lance Armstrong, you'll probably know the story of Lance Armstrong with testicular cancer. He was cured with conventional chemotherapy used in combination, but again, the discovery of those drugs was not informed by the knowledge of which genes were actually altered in, for example, testicular cancer. So where we need to get is that for every cancer, we have what we're referred to as precision drugs, where this is the genetic abnormality, and if you have this genetic abnormality, this is the right drug for the cancer. And think of like a key and a lock, the drug being the key and the abnormal gene product being the lock. In colon cancer, we're not quite there yet, but just as an example of how progress is coming, the majority of colon cancers have abnormalities in a gene called RAS that Jonathan referred to. And RAS is overactive. It's like an accelerator in these colon cancers. So the dream for my entire career has been, wouldn't it be great if you had a drug that turned off RAS? And people threw up their hands and said it's impossible, it doesn't have the right kind of properties, you'll never get a key to fit in that lock. But now, in the past year or so, we have the first RAS inhibitors showing activity, and there's a next generation that looks even better behind them. So you do the best you can with the drugs that are available at the time, and for some patients that does involve standard chemotherapy. But increasingly, for many patients, we're able to use these more targeted precision drugs, which typically have fewer side effects than conventional chemotherapy.
I would say conventional chemotherapy is probably not going to go away completely, because one of the things we've begun to appreciate is some conventional therapies, which you might think of as completely blunt, are actually a bit targeted in their own way, depending on the patient. As Bill said, at the time they developed, nobody had the slightest clue. They were basically throwing chemicals at cells and tumors and seeing what worked. And, if it works, it works. But you think of even things like tamoxifen. It's basically is a targeted therapy. It was developed a long time ago. It's not really a chemo in the way you're mentioning, but some of them do have some targeting ability. I mean, certainly they can discriminate between rapidly growing cells and ones that aren't rapidly growing. That was the basis for their selection in the first place. So it's not, I don't think, and maybe there'll come a time when they're completely out of the picture, but I think at least for the next decade, it's going to be combinations of conventional plus targeted plus immune or epigenetic or some combination thereof. As Bill mentioned, the more nodes you can attack, the more chance you have of suppressing it completely.
Was there a question?
We have a question here.
Yes, you've mentioned a number of new therapies that have been recently, in the last 10 years or so, come up. Can you comment on the utility, perhaps, of gene therapy and CRISPR or other gene editing technologies for the treatment of cancer?
So CRISPR is, I would say, in the laboratory becoming a godsend, because if cancer is a problem, which it is, where specific genes have been altered or mutated, and what you'd really like to have in the laboratory is the ability to then rewrite that genetic sequence. So maybe what would happen if you corrected the sequence, or alternatively, what would happen if gene by gene by gene by gene, I use CRISPR to turn off the different genes, gene A, gene B, gene C, until I found the gene that, when inactivated, killed that colon cancer cell, for example. And that then serves as the first sort of piece of evidence that you might want to have before you go to our friends in pharma to say, we found it. This is the Achilles heel of this type of cancer. So already we use CRISPR to interrogate the functions of the genes. We have the technologies now where we can actually interrogate all 20,000 genes in one experiment using CRISPR to toggle them all on or off in parallel. Then the other thing we're doing now increasingly is, if we know a common cancer is caused by mutations in gene A, B, or C, but in combination, we can now use CRISPR to go into a mouse and affect gene A, B, and C, and the mouse gets that cancer. So now, before we test the concept in a human being, we can test the concept in a mouse that has precisely the same genetic abnormalities that we know will be important for the patients.
Yeah, I think what the public in general is expecting from CRISPR is what I would expect too, is that if you have the ability to fix a mutation, why don't you just go and fix it? And then the cancer would be done with. And it's probably true at some level, but the engineering issue is not trivial at all, because unlike some diseases where you're trying to restore something, let's say you have a clotting disorder and you use CRISPR to restore a little bit of your clotting factors. A little bit's probably enough. Or sickle cell anemia, they just approved, I think there's a treatment now using CRISPR that essentially restores enough regular hemoglobin that you don't suffer from sickle cell. Those are gain of function. But in cancer, a lot of times you're trying to disable something. And so if you don't hit all of the targets, like you have 1 trillion cells and you fix 500 billion, well, you still have 500 billion that aren't fixed. So conceptually, you should be able to do it. Like, if you can fix them, you can fix them. But engineering-wise, that's a very formidable problem. And so its application to common tumor types as a treatment, probably a bit off.
Yeah. Probably the first applicant, I completely agree with that. So that is the engineering challenge, that until you have a technology where you can go into every cancer cell in the tumor and correct every gene, we're not going to get there for the reasons Jonathan stated. But what we are already seeing are examples, and some of this work's being done in Philadelphia, is taking CRISPR and re-engineering the immune cells to make them even better immune cells at killing cancers. So I think that'll probably be one of the earliest applications of CRISPR for cancer therapy.
I think we're going to take the question there first, and then we'll come to the front.
Being naive, what is CRISPR? Could you explain that?
We'll let the Nobel Prize winner.
Thank you, and you're also polite because there were a couple times during our discussion where scientists are so terrible slipping into their own private jargon that I wondered whether we might have also been guilty a few other occasions of using this terrible jargon that we use. So what is CRISPR? So one of the reasons I love the story of CRISPR, before I tell you what it is exactly, is the people who made the basic discoveries that enabled CRISPR, which is a tool for gene editing, and we're going to get into it a little bit more, weren't actually thinking they were working on cancer. They didn't even think they were working on human disease. They were just following their curiosity. They were seeing some very peculiar things happening in the laboratory where it seemed that in some cases bacteria had effectively figured out a way to become immune to certain viruses. And so they were just following their curiosity. But that's where a lot of big transformative discoveries come from. In fact, that work did lead to the Nobel Prize 2 years ago because it's a complete game changer. But CRISPR is an acronym, and even I don't remember each letter and what it stands for. But in the end of the day, it's a very elaborate system that organisms have where if a foreign DNA enters the cell in a very precise and targeted way, the host cell can cut that DNA and disable it. But once you started to see the pieces of the system working, a very smart scientist named Feng Zhang at MIT figured out, well, if E. coli can do this, if bacteria can do this, with a few tweaks, we can do the same thing in a mammalian cell, including a human cell. We can take the basic building blocks of this system and now re-engineer it so that we can cut a genome and then use a template that would be used to change the sequence in a very precise way. So I'm sorry I don't remember what the acronym. R is repeats, P is palindromic, C is clustered. I'm almost there. Okay, interspersed. I get a B. I think I get 4 out of 5.
I would like to just add on to that story. So Feng Zhang, who Bill just described at MIT, actually was funded by Damon Runyon to do a bunch of his early work. And so we were in the room. This was 2011, I believe. And we were sitting in the room. He gave a presentation as part of the finalist interview to our scientific committee. And he gave his, 10-minute presentation, and we had an opportunity to ask him questions for another 20 minutes or so. He left the room, and everybody looked at each other and said, God, if this actually works, this is going to actually change. Exactly. And so it was amazing to be there at the very start and to be able to support.
Yes, it seems like every 10 years or so, in the last at least 30 or 40 years, there's been some revolution of this type. I mean, right before, 10 years earlier, there was something called RNA interference that came out, which was also revolutionary for its time. It's been a bit eclipsed by CRISPR now, but it also was a game changer. And before that, sequencing and things like that going back in the past.
Was there a question?
We have a question in the middle row and in the front.
Who's
Lisa,
next?
if you want to take this one after. All right, we'll take the one there first.
So thank you all. This is a really fascinating panel. So I work in the field of cancer immunotherapy, and I'm curious your thoughts on why we haven't seen more of the combinations be successful, bispecifics, other things, since we had the first CTLA-4 and PD-1 approvals, why we haven't maybe made more progress with combinations, et cetera.
I feel like I'm...
I don't know.
Well, so it turns out in general, and by the way, in the interest of full disclosure, I'm on the board of directors for Lilly Pharmaceuticals, and I also consult for a number of other biotech companies. If you look at drug discovery across all therapeutic areas, not just cancer, if you have a target that you're pursuing where there's what we call genetic validation that this target really, really, really is causally linked to this thing you want to accomplish, that increases your probability of success dramatically. And then secondly, your chance of succeeding goes even higher if you have some biological understanding of what this target actually does. So for example, some of you may know that there are human beings walking around on the planet that have actual genetic alterations they were born with in a gene called PCSK9. And depending on the nature of the mutation, your cholesterol goes high or your cholesterol goes low. So that's the definitive genetic smoking gun that if you want to manipulate cholesterol levels, maybe you should think about going after PCSK9. If you had a drug that made it more or less active, it would reasonably be thought that it would change cholesterol. And moreover, we knew enough about what PCSK9 did biochemically to understand why alterations in this gene would alter cholesterol. So now you go back to immunotherapy. So the targets that have worked really, really well in cancer are targets where if you make a mouse with a mutation in that gene, the immune system ramps up tremendously. So that told us that these genes really were rheostats on the immune system, and that if you could inhibit these, you'd amp up the immune response. And then it took a person like a Jim Allison to say, well, if that's what you do, maybe this would be a way to treat cancer, because I want to ramp up the immune response. So it turns out for some of these other targets that are being pursued, the genetic evidence and the biological understanding, I would say, is not quite as good as it was for CTLA-4 and PD-1 and PD-L1. But, let's be candid. Our knowledge of biology is still incomplete. So there's still a lot of guesswork involved in developing drugs. So I think a lot of these other targets that were pursued were very good guesses based on the knowledge that we have, but our knowledge was simply incomplete. And the final thing I'll say is I think the immune system is one place in particular where mouse models are often inadequate. You don't really know what you're going to accomplish until you do the clinical trial. I'm probably speaking to the choir here.
I want to answer this question of the tumor immunologists in the room, but maybe for everybody else, and that is that, there was this revolutionary change from 5% response rates or 10% response rates with some conventional therapies and, stage 1 immunotherapies to now 30, 40, 50%. And that's not a home run. And as I mentioned before, that's 2 or 3x. But there's a problem with overlapping toxicities. There's a problem with the maximal tolerated doses, as you well know. We talk about, should we do them in sequence? Should you get immunotherapy 1 followed by immunotherapy 2? Should you get immunotherapy 1 with immunotherapy 2? How do you put them in the context of someone who just had major surgery? Most people know that immunotherapies, while they're actually quite effective in many different tumor types, melanoma, lung cancer, kidney cancer, a lot of the liquid tumors, et cetera, they don't work in other tumors. They don't really work very well in prostate cancer. They don't work very well, in breast cancer. I mean, there probably are segments of people that we haven't yet identified who would be better responders, but as a whole, those are more driven by endocrine features than they are by immunological features. My point being, though, that, when you take these immunotherapies, there are autoimmunities that sort of limit your ability to keep giving it to people, whether it's, autoimmune colitis or thyroiditis or pneumonitis, which can sometimes be actually, fairly so toxic that they're lethal. And so I think that, as was mentioned before, when it sort of shows up in a clinical trial for tumor type 1, suddenly everybody starts to use it for the various tumor types. And frankly, the tolerability in those settings is sometimes the limiting step.
Thank you. We'll take one final question.
You mentioned HPV. Could you touch on vaccinations or other preventions that are being worked on or we should know about?
Does anyone want to speak?
I'll start a little bit with this one. I think most people don't really understand fully the relationship between, oncoviruses and cancer, right? So it was mentioned that, there's this ability for, external DNA to be inserted into your own DNA, and that's a very common technique that viruses have. They insert their own DNA into, human DNA, and sometimes those can drive, cell cycle to sort of, cause this proliferation of cells and cause cancer. I mean, that is the definition. So if you can prevent the infection of the disease to begin with, then of course you can perhaps potentially affect the incidence of the cancer. That was shown, as I mentioned before, with hepatitis and the liver cancer virus and hepatomas. And it is a very effective therapy. And I think people they probably underappreciate the role of vaccination as a preventative form of cancer. And it's probably because, we haven't yet proven things like prostate cancer are virally driven. And breast cancer, we don't know those types of things, but liver cancer is, Burkitt's lymphoma is, head and neck cancers are, cervical cancers are. And so that is an excellent way to prevent cancer, and that is vaccination. I don't know if people have other thoughts.
We happened to work at the institute where Barry Blumberg was, so, discover hepatitis B. And I remember, at his funeral, his son-in-law talking about the vaccine that he created, prevented tens of millions of cases of liver cancer in Asia and other places, and that because of that, global lifespan increased 8 months or something. You think about impact. I mean, to change global lifespan over billions of people, that's tough. And that similar argument could be made. I don't know the precise numbers for HPV viruses. That by preventing those cancers, they're doing far more than we'll ever do with therapies for cancers that have already developed. And now we're beginning to enter an age with the new types of vaccine. Pancreatic cancer vaccine was mentioned at the very beginning, and others. I think if those work anywhere near as well as these antiviral agents have done in prevention, that would be a true revolution and might make our treatments obsolete because they wouldn't be necessary.
And if I may, I could do a medical student trick here and answer a related question that I'm more equipped to answer than your question, but it does relate, in fact, to the previous question. So, we've been talking a lot about the genome. You probably all know that your DNA is made up of 4 different building blocks called bases, and scientists represent them with 4 letters, A, G, C, T. And a human being has about 3 billion of these. So when we talk about the human genome, we're talking about these 3 billion bases, the 4 building blocks, and the actual sequence determines that we're here as human beings. But it turns out, again, through basic science, we now know that about 8% of your genome is not really human. It's remnants of viruses that have infected Homo sapiens over the course of evolution. Now, fortunately for us, most of these viruses are not intact at all. A sophisticated scientist could see this little sequence here is from a virus. It's not really human. But the other thing we're learning, and I'm looking at Dr. Celeste Simon, we're also learning that in some cancers, some of these remnants or pieces can be re-expressed. And in some cases, by that I mean that the gene gets activated and its protein product gets made. In some cases, it's looking like the immune system can see that. So when the immune system sees a cancer, in some cases, it's clearly seeing that this cancer had a mutation in those genes that allowed it to be a cancer, or in some cases, maybe it was an innocent bystander gene that got mutated, and that's what it's seeing. But we're increasingly thinking that in some cases, the immune system is seeing this inappropriate re-expression of viral genes and viral proteins, which is good because now that we're learning the rules, we think we can turn those things on or off to make the cancers more easily recognized.
Thank you all so much. So in the interest of time, unfortunately, we are going to stop there. I want to thank all of our panelists for a very fascinating and engaging discussion this evening. Thank you. Oh. Hold on. Wait, one more.
I'm going to paraphrase President Kennedy when he introduced his cabinet. He said, it's the greatest collection of minds since Thomas Jefferson dined alone. We have the greatest collection of minds here that I've ever seen. They are fantastic, and I just can't thank them enough for coming. And please join us for dinner. It's not just cocktails, it's dinners in the Franklin Hall. So thank you so much for coming. I really appreciate it.
Thank you.